Hello and welcome back. The topic for this video now is going to be cloning, all right? And in the format that this is in, being online, it's a little more difficult for me to start a discussion with cloning related to the ethical considerations associated with it. And there certainly are some ethical considerations to be discussed when talking about cloning. What I'm going to focus on in the video is going to be how cloning occurs, all right? How the clones are actually made, all right? But don't think I'm ignoring the ethical implications here, because I'm not, okay? When we're talking about cloning, we're talking about making an exact genetic copy. So when we talked about mitosis and we talked about the clones that came out, the process of mitosis in cell division, you are generating a clone of another cell. You've got an exact genetic copy. The DNA in one cell is the same as the DNA in the other cell, okay? And the concept of cloning has been going on for centuries, although there's been a lot more about it in the news in recent years. What we're talking about when you're making an exact genetic copy, an example of where you may have done this is if you've ever given cuttings with plants and given that to a friend. You are essentially giving them an exact copy of the plant that you have, all right? They're growing another plant that's going to have the same DNA makeup as the plant that you have, okay? So conceptually, it's not a new topic, but what makes this more controversial is when we start talking about reproductive cloning. This is where the ethical issues come into play, okay? So we're talking about making an exact genetic copy of a particular organism. And the organisms that we're talking about, if you're talking human, dog, cat, things like that, okay? Now, in terms of reproductive cloning, there have been some successes associated with that. Back in the 1990s, there was Dolly, the sheep, okay? Dolly was a clone of her mother, all right? She had all the same DNA as her mom did. So it was an exact copy, all right? Now, with cloning, with reproductive cloning, there are some caveats that go along with that, okay? And in terms of those caveats, the clone tends to have a shorter lifespan. So the clone doesn't live as long, okay? Typically, with the type of sheep that Dolly was, a normal lifespan for that sheep might have been 11 or 12 years. In Dolly's case, she lived about 6 or 7, I don't remember. So about half of what would be expected, all right? And in terms of Dolly, there were also some health concerns that came up. Now, whether that's something that would have normally arisen or not is hard to say, because they only had the one sheep to make these conclusions off of, all right? But what Dolly was able to do, she was able to reproduce, we'll call this normally, all right? But what I'm referring to here is she was able to, given sexual reproduction, was able to have offspring as a clone, okay? So her offspring would not have been genetically identical to her because it was the result of sexual reproduction, all right? Cloning is an example of asexual reproduction, okay? So just keep that in mind, all right? And essentially what they did, okay, I'm going to draw a cell here in green, and I'm going to use, should we use pink for Dolly? Let's use pink for Dolly, okay? This is going to be an unfertilized egg. Essentially, what they did in order to make Dolly was the process of nuclear transplantation. So the cell they took from Dolly's mom was a cell from the udder. So in anatomical terms, it came from the mammary gland, okay, or breast tissue, okay. What they did is they removed the nucleus from the mammary gland cell. They removed the nucleus from the unfertilized egg, and they transplanted the nucleus from the mammary gland. Because what we have here, this is going to be 2N in DNA content. I'm going to draw our egg over here again. This is going to be 1N in DNA content. That's why that nucleus was removed. We're inserting a nucleus in now that is 2N, so it has a complete set of information to make the individual. And what happened, and what they needed to kind of get that to start dividing, they provided a bit of electrical current to that egg once they transplanted that nucleus. And that started that cell dividing, okay? So one cell divided into two, two cells divide into four, and in transplanting that nucleus, there was something in the DNA that was reset, okay? It reset the programming on that nucleus so that it thought it was an embryo, okay? Those cell divisions happened, all right, and these cell divisions were followed in a petri dish, so they were followed in the lab until that embryo got to what's known as, let's give them all little green nuclei, right? Until that embryo divided enough time to become a blastocyst. Once it became a blastocyst, it was then transplanted into a surrogate mom, where that blastocyst could develop into, in the case of Dolly, a sheep, all right? Now, in terms of cloning, when I talk about it in face-to-face classes, one of the things I like to ask is, how many times do you think they needed to do this to get Dolly, all right? And I get a range of guesses, but in terms of this, I'm just going to jump to the conclusion here since you can't give me your answer, all right? It took 264 tries to get one live sheep. So the thing to take away about cloning, this is not terribly efficient. It's not an efficient way to reproduce, okay? And the other thing that's interesting, I think from a cloning perspective, is that there have been successes with some species. So Dolly being cloned was huge because that was a large vertebrate animal that showed that cloning could happen. There have been attempts to clone cats that have been successful. There have been attempts to clone dogs that are also successful. Interestingly, monkeys, which are most closely related to humans, have been less successful. And then this gets into the ethical part about cloning and humans. All right, in the United States, labs can't take this process and have it be human eggs and that are used for the process of reproductive cloning. All right, there's a ban on that. So in terms of the cloning, I think it's interesting that some species it's been relatively easy to get clones out, and other species it hasn't. I think that has some implications about the DNA that makes up those different organisms, all right? And there may be modifications that go with that, all right? Now, in terms of that reproductive cloning, what makes it reproductive cloning is taking it to the point where it gets transplanted into a surrogate and host, a surrogate mom, and you get a live organism out, okay? So that's what makes reproductive cloning, all right? There is also the concept of therapeutic cloning, and that's what I'm going to talk about in the next video, all right? So hang on, there's more to go with cloning. If you have questions about this, please let me know.